gentlemen, I'm absolutely delighted to be speaking to you here in the City of London, not because it's the home of the investment world, but because it is more or less equidistant between Bart's Hospital and the Royal London Hospital. And right as we speak now, Bart's Hospital is doing important work with the Angle Parsortix system in prostate cancer. I'm also very pleased to see some existing shareholders in the room and some familiar faces. I hope that you will get some interesting update from tonight, and certainly you can come and quiz me afterwards on the questions. I'm equally happy to see new faces, people I haven't seen before, because I want to tell you about a tremendous opportunity. We believe that Angle has a very, very simple idea, an approach which we own with patented technology, which is going to revolutionize and transform the way that people are treated for cancer. And that's going to have a number of major impacts. It's going to reduce the medical problems from cancer. It's going to improve people's lives who have cancer. It's going to reduce healthcare costs and therefore make the burden of other treatment easier to carry. And importantly, that is going to translate into driving shareholder value for Angle's shareholder base. And what I'm going to talk to you about is personalised cancer care. This is about what happens to an individual when they have cancer. And it is an assumed thing that if somebody has, for example, breast cancer, then they have the same cancer as another person who has breast cancer. That is just not the case. It is not the case at all. In fact, it is simply that they have cancer in the same part of the body. And this is exactly the same with colorectal cancer, prostate cancer, and all the different cancers. This is a disease which is very heterogeneous. The cells in the cancer disease are different from one patient to another. And the real problem that we're suffering at the moment is that everybody is treated with a one-size-fits-all approach to care. So they're all offered the same treatment even though their diseases are different. That seems crazy, doesn't it? I'm going to explain to you why that is, and I'm going to explain to you how our company with our product called Parsortix is going to help to effect a change to deliver what Barack Obama calls precision medicine. That is medicine targeted to the disease of the individual patient. That is avoiding giving patients treatment which is not helping them, it's giving them toxic side effects, and it's costing a lot of money. So far, far better to identify what specific treatment will help that individual patient's disease. But it's a monumentally difficult task, though, because not only does each patient have a different disease, even though they've got colorectal cancer, they've all got different diseases. Not only that, but their own disease is constantly mutating and changing. So the doctors may have an idea initially, but subsequently during their treatment, they don't actually know how to treat them. And obviously, we've got a big problem with cancer. This slide's actually now out of date. Cancer Research UK have published statistics saying that 1 in 2 of us in this room is likely to experience cancer during our lifetime. So it's a very, very prevalent disease, and unfortunately a lot of people are going to be dying of that in the future. But in terms of the need to reduce healthcare costs, I've touched on that already. The way to do that is to provide specific drugs and other therapies that actually meet the specific needs of the individual. So if you could identify which mutation that patient has and then give them a drug which helps them, you're reducing costs and improving their outcome. And listed there are a number of the major drugs which have been developed which do just this. Every single major pharmaceutical company in the world is developing targeted medicines for cancer at the moment. And every single one of them needs to understand the mutational status of the cohorts of patients that they want to treat. How do they do that? Well, the standard way of doing it is something called a solid biopsy. And this is simply a surgical extraction of some of the cancer, cutting out some of the cancer so that those cancer cells can be analysed. And that's standard. And you can imagine in breast cancer, there's a lump. The lump is cut out. That's how they get the cancer cells. Same thing with colorectal cancer. They use a colonoscopy. They find a growth. They cut that out and they analyse it. And that gives a good bit of information and enables the selection of drugs at the start of people's treatment. However, how many times have we heard of people we know whose tumour has reduced? We've been told everything's been very, very successful, a 90% reduction in the tumour, and everybody, including the patient's hopes, rise, only to be dashed when we hear that the 10% that's remaining has not been eradicated, the drugs that are being deployed are not working, and the patient is unfortunately going to die fairly soon. And this is because the remaining 10% is mutationally different from the 90% that's being cured. It is absolutely essential to understand the mutational status of the remaining cancer during the course of the patient's treatment. But there's no way to do that, because you can't go back in and cut out the lump. You've already cut the lump out, it's no longer there. So how do you get cancer cells? Well, very fortunately, with regard to our product and with regard to the treatment of patients, there are circulating tumour cells which exist within the bloodstream. And what happens is the primary tumour is growing and it drops cancer cells into the bloodstream. Now, these are pernicious metastatic cells which are seeking to find somewhere else in the body to land and start a secondary cancer. And most patients die of their secondary cancer. However, they offer the key to this particular puzzle. If you could get hold of those cancer cells in the bloodstream, then you would have a ready, easy, repeatable, non-invasive way of getting hold of the cancer cells to check and work out which drug is going to be most effective for that patient. However, it is a phenomenally difficult technical challenge. And you'll see that the engineering solution that we have developed to address this is very, very smart. I would point out at this point, this is technology which is owned by Angle plc. We have intellectual property covering this worldwide, including 2 granted US patents. So, this is going to show you how our system works. We have a small microscope slide shaped cassette. I'm going to hold it up in a minute, but first of all, just watch this animation. The blood flows into the cassette, and this is just showing you an illustration of how it works. We have a series of steps which are designed to capture the cancer cells, which are shown green here. The white blood cells obviously white and the red blood cells red. You can see the cancer cells are much larger than the other cells. They're approximately 3 times bigger than the white blood cells. So we have a patented gap system whereby the gap size is set at a level which will capture the cancer cells. Everything else will wash through. There'll be a few residual white blood cells and a few red blood cells, but basically it's the captured cancer cells, notwithstanding the fact that there's only 1 cancer cell in 1,000 million blood cells. This works. And I have one in my hand here. So there's a microscope slide plastic consumable. This is a one-time use piece of kit. It is nano-manufactured to plus or minus 1 micron, so it is done in a very precise way. The design of it obviously took us a lot of effort and years of investment, but now it's here, and it's made by a plastic moulding system. It has enormous value, but it is low cost. That's the key to a successful financial outcome. I'm now going to show you what it looks like when blood actually runs inside one of those cassettes. And that's just going to come up. Now what you see there is the dense red colour of the red blood cells massing up as they go over the step. And the steps here are these lines. That's looking on top, that's why you see a line. And what I really want you to see here is the easy flow of the blood through this. It doesn't clog up, it flows straight through. It's light coloured in the middle because that's where it's a constricted gap and there's not so many cells there. And in a minute, you're about to see a trapped cancer cell. So this is 1 in 1,000 million cells, which just happens to be a large cancer cell. And there it is sitting there, captured. It's not moving, but it doesn't affect the flow in the rest of this cassette. The blood flows right past it and disappears. And you end up with the very large captured cancer cells there. The gap between here and here, by the way, is 20 microns in distance. Whereas the white blood cells are more like about 5 microns. So that's how you can see these are very big cells. Okay, so we've got a machine which drives this. Very simple machine. It's a series of pumps and valves. It's been precision engineered, but basically there's nothing particularly clever in there other than the way that it operates, which is fully automated, so that the user simply loads a blood sample from the patient. Now, just to be clear, this is a peripheral blood draw, just exactly the same as probably everybody in this room has had a blood test sometime in their life. That's all we're talking about. Very simple. They take that exact tube, take the top off it, put it in our machine, hit the button, and it's an automated process which pushes the blood through the cassette that sits inside that purpose-built clamp. So the user doesn't have to do anything except load the blood and hit the button. When it's finished, they can change this valve and then they can run the harvest procedure, and that very cleverly reverses the flow washes the captured cells back out. All the blood cells have gone to waste, washes the captured cancer cells out and presents them for analysis. So we're calling this a liquid biopsy. It's no different from surgically cutting out material to get cancer cells. It's just easier to do and can be repeated as often as you might like. So obviously this has a transformational potential for treating cancer patients, and my job is to get this being used in treating patients as fast as humanly possible. We've estimated there's an £8 billion market for this, so it's a huge market opportunity. As you can imagine, there are multiple competitors who are seeking to address the same market. There are about 20 that I'm tracking. One of them is a very large company. It's part of Johnson & Johnson, and that's got a product called Cell Search, which is the first one. The top line here represents the attributes needed, we believe, for a successful system, and as you can see, our system, Parsortix, meets every single one of those. However, the only FDA-authorized one, the Johnson & Johnson one, doesn't. So the key attributes of the system are: can it work for all types of cancer? Answer, yes. Can it work for all types of cancer cells in the bloodstream, including the mesenchymal ones? They're the ones that do the metastatic secondary cancers. Answer, yes. And can it easily harvest the cells out of the system for analysis? Answer, yes. And can it provide cells with high purity? What I mean by high purity is low background white blood cells. You want to get as pure as possible cancer cells and less of the blood cells in there, and yes is the answer to that as well. So this is a system that's been fully developed and it's working. So now we're taking it to market, and we're moving as hard and fast as we can into the sales cycle. The concept's developed, as you've seen. It's been productized already. And we've got KOLs, that's key opinion leaders, major cancer centres. I mentioned Bart's right at the beginning of this presentation. I'm going to show you some others. We already have 5 major cancer centres who have evaluated this system. There is absolutely no question that it works in capturing cancer cells, and it presents those cells in a form that can be analysed. So, of course, we're now involved in translational research. The key thing is getting the big cancer centres to use this device in order to treat patients and get better outcomes for those patients. And we have 9 major cancer centres around the world who are working with our system on doing that right now. You can't sell something for clinical purposes unless you've got regulatory authorization. We already have this. This is approval for use in treating patients. We already have this in Europe in the form of the CE mark, and we are hard at work seeking to get FDA approval for this system. We're already 13 months into a process with the FDA. As far as we're aware, we're the only company that's engaged with the FDA, and we hope to be the first FDA-authorized system in the world for harvesting cancer cells for analysis in the way that I've been describing. I'll come back to the corporate deals and the research use immediately. There are 5 big cancer centres, they're shown here on this list, who have already evaluated our system and confirmed that it works. And not only does it work, it has major advantages over other systems which are being developed. And I've covered some of those already. And in addition to this, there's another 3 down here that are working with the system. And there's a 9th one since this slide was produced, which is MD Anderson, arguably the biggest and most successful cancer research centre in the world, based out of Houston, Texas, who are working with our system in colorectal cancer. Right, I said we were moving into the commercialization phase, and we've made a commitment to the market that we will be generating first sales from this product in the second half of this calendar year. That will be for research use. So now that we've got major research centres who say this works very well and they've been using it, we're starting to sell to other parties. We've got some big commercial advantages. If you look at the pricing on the right-hand side here, the pricing that we can price at is well ahead of the cost base. And actually, we can price our product below the cost of our major competition. So we've got the cost side completely sorted out. In terms of research use, the main big users we see of this is the big pharmaceutical drug companies. And we offer them the ability to reduce the time for developing new pharmaceutical drug trials, to reduce the cost of those trials, and most importantly, to actually enable them to assess which people will benefit from their drug and which won't. And that's becoming a crucial factor in getting regulatory approval for new cancer drugs, because these drugs are so expensive. £50,000 is about a baseline cost for an individual course of a cancer drug. And some of the big new drugs are £150,000. Well, no health service in the world wants to spend that kind of money and find that the drug's not being effective. And very often these drugs only work for 1 in 3 people. But the question is, which are the 1 in 3? So we've worked out that there are 750 phase 2 cancer drug trials open to us and suitable for our system every single year. Each one of those can generate £100,000 of revenue for Angle. And there's 120 much larger scale phase 3 trials. Each one of those might generate £750,000 to Angle per study. So our estimate is that at 5% of that available research use only market, we could generate £8 million of revenues. I've already shown you the margins. Just by reference, our cost base historically has been below £4 million. So that's potentially double what our cost base has been historically. So that's the research use market. That's available to us right now, and we are beginning to sell into it as we speak. However, the big prize is the £8 billion market, which is sales for clinical use. I've already talked about the size of this market and the regulatory authorization. The big thing here is that we've got multiple key opinion leaders. These are the world-leading cancer research centres that you've seen working on the translational research to prove this. So they are consenting patients literally as we speak and putting them into clinical studies with our Parsortix system to show how well it works. The first one of the key opinion leaders to report is the Medical University of Vienna. Now, the Medical University of Vienna is arguably the leading ovarian cancer research centre in the world. And their team has been working with our Parsortix system in treating ovarian cancer. Now, ovarian cancer is often named the silent killer. And the reason it's named as the silent killer is, very unfortunately, it's difficult to identify ovarian cancer until it's very late. And if a patient is identified at stage 4 cancer, then I'm afraid she only has 3.5% chance of being alive 5 years later. It is a real nasty killer. However, if she could be identified at stage 1, then she has about a 90% chance of being alive in 5 years. Obviously, it's incredibly important to try to identify ovarian cancer. I find it shocking that there is no way of doing that which is successful at the moment. There is no way. And women who have abnormal pelvic masses, which is a growth which has been identified, so they've got symptoms, it's been identified, they've got an abnormal growth. They're submitted to surgery to cut that growth out without knowing whether they have ovarian cancer or not. And if they have ovarian cancer, they need a specialist oncological surgeon who's experienced to do a 6-hour operation to cut that out. And even with that, their circumstances are very difficult. And that's about 11% of the women who, there's probably about 500,000 women a year in the United States and Europe who have these operations. 11% of those will have ovarian cancer. The remaining 90%, however, will be benign, and probably they just need a general surgeon, and it'll take about an hour, and it's relatively straightforward, comparatively straightforward. So obviously you need to know who are the ones with ovarian cancer. They need an intensive care bed waiting for them. They need an anaesthetist who knows what they're doing, is ready for 6-hour operation. If they don't have that, their outcome is terrible. Well, that's what we're targeting as our first application. The Medical University of Vienna people have been working on the identification of ovarian cancer. They've got some very clever RNA markers which can accurately discriminate ovarian cancer cells from any other cells. And so what they need to do is to capture the circulating tumour cells, the cancer cells from the bloodstream. They've worked with substantially every single system available on the market. None of them has done better than a 24.5% sensitivity. So that means that 4 patients who had ovarian cancer, they only correctly identified 1. In our system, in the initial study, they've identified 90% sensitivity. And what's more, they had 100% specificity. The biggest problem with the existing systems is the specificity. That means false positives. So how many times does it say somebody's got ovarian cancer when they haven't? The answer with current systems is each one they get right, they get 1.5 wrong. Well, that's not a usable system. In our case, 100% specificity. In healthy, normal volunteer patients, they didn't find any which showed up as if they had ovarian cancer. And Medical University of Vienna, I'm very pleased to say that as part of our work in the United States, we're participating in AACR, which is a massive cancer conference next week, and there will be a poster publication from Medical University of Vienna giving a lot more detail on this product. So, having got these excellent results, Angle has engaged, and the fundraising we've done earlier this year is being deployed specifically to get clinical studies to basically substantiate these results so that we can go ahead and start providing this care to ovarian cancer patients. And that market alone is worth £300 million in revenue per annum to Angle. Meantime, we've got, Ian, you want to get ready, we're nearly into questions. Meantime, we've got multiple other clinical applications that I've mentioned. We've got some people in the United States here. This is a leading cancer centre there, specialising in metastatic breast cancer. That in itself is a £750 million market. I mentioned prostate cancer with Barts, which is nearby. Cancer Research UK Manchester, I haven't dwelled on them, but they've been working with our system for over 2 years now. The Manchester Institute is attached to the Christie Hospital in Manchester, which is Europe's largest cancer hospital by number of patients. And they're working on colorectal cancer and pancreatic cancer. So there's a whole wide range of applications that we can roll out in Angle. And so just to summarise now, and then we'll move into questions, what we're talking about here is a simple blood test. Nothing more complicated than that. We're using an engineering solution to a very, very complicated technical problem, but the solution we're using is very simple. The system has been developed already. It works already. 5 big cancer centres have confirmed that. And we now have 9 major cancer centres doing translational research, who are all people who are the top of their game in the cancer research fields, whose job is to try and bring treatment to patients. We've got a clear path to commercialisation. We're moving into research use sales this year. Shortly, we're going to be, as soon as possible, going into clinical sales, probably about 18 months from now. We've got funding in place, having had an oversubscribed placing earlier this year. Angle's got a lot of momentum building. We're looking to make Parsortix inside. So basically, we're stealing this term from Intel. If you look at every laptop and computer, it says Intel inside. Intel inside. Doesn't matter which manufacturer it is. That's what we want. We want to have Parsortix inside all of the analytical systems which already exist for analysing cancer cells. We're just giving them the cancer cells in a different way. Thank you very much.
Thank you, Andrew. Do we have any questions? Any hands up?
Thank you, Andrew. I note we share our surname, which is a good start. Inspiring. How far off are you from full FDA approval in terms of cost and time post-clinical trials?
Yes. So the FDA authorization is something that, as I mentioned, we've been working on for longer than that, but we've made a submission to them 13 months ago. And we're in an active dialogue with them to get our system approved. I'm standing with a light in my eyes. Get our system approved. We presented them with some challenges. The first challenge is we want to be the first system that has ever been approved for harvesting cancer cells. And that in itself creates a number of sort of concerns for them. They're trying to work out what questions they need to ask us and make sure we're giving them the correct answers. Predominantly to do with health and safety and outcome for patients. The second problem is if you like, a self-invoked problem by Angle, which is that we have deliberately gone for a wide application. So we've said we want to get an approval for any form of cancer cell extraction and then any form of analysis once we've got the cancer cells out. And that creates problems for them because they're much more used to doing very, very targeted. If you just do this about this particular type of patient and this particular indication, what happens? So we're challenging the FDA. We're doing that deliberately because what we want to do is get an authorization which allows wide use of the system. In terms of, do you want to comment on costs at all?
Pursuing the FDA is a relatively modest cost, depending on what they come back on some of the data. The actual regulatory advice and the external advisors we're using, it's modest. We've put data in, we've got a lot of data. where there might be a cost implication if they come back and ask for some additional data, but we're working with the key opinion leaders to get that data in any case because we need it for clinical applications further down the line.
Thank you.
Any other questions? Can you wait for the mic? Thank you.
I note you only need a CE mark to sell into Europe, which is great news. In terms of manufacturing the machines and the disposables, what's your plan for manufacturing those, and where are you gearing up?
So we've taken a very lean approach to how we develop the business. So we've outsourced our manufacturing and a lot of the design that was associated with getting the product development and the cassette developed. So in terms of the machines, we use a company called Cogent, who are based in Suffolk, and they're a pretty large organisation. We can scale it very quickly. As Andrew mentioned, we use a lot of standard components, so there's not long lead times, so we can turn the handle fairly quickly. The underlying cost, it's about 6.5K for a machine, so it's very, very easy to do. In terms of the cassettes, they're a slightly longer lead time. It's a specialist nano manufacturer based in Germany called Thinks. But again, we just have to get into their workflows, but they're also geared up. They can scale up and produce as many cassettes as we need. So we have no limitations in terms of machines or cassettes and what we can produce and when we need it. It's just a fairly modest lead time.
Question at the back, please.
Sorry, Andrew, could you please give us a little bit of background of yourself? And second question, who are your competitors in this?
Yes, okay, myself. My first degree is in engineering science at Cambridge. I worked in some engineering companies during my gap year and then in my long vax. I realised that there weren't any engineers running these companies, and what I actually needed is some commercial experience. So I trained in finance as well, so I'm a chartered accountant. I went with KPMG. I worked for a couple of years in the United States, in Boston, Massachusetts, with the emerging technology companies. I actually did one of the first ever floats for a medtech company in Boston while I was there. Came back, set up the technology practice at KPMG in UK and then Europe, which is where I first started working with Ian. We were at KPMG together, and then founded Angle in 1994. We've had a history where we've established a number of technology companies. It's all to a 25-year plan, and that was to identify one really world-class company and focus all our efforts. So those shareholders in the room who have been following us for a few years will have seen that we have progressively over the last 3 years, we've sold out of various other companies and reorientated all our resources into Parsortix, because this is the best opportunity we've come across ever. Oh, you wanted to ask about competitors? Sorry. Competitors, there are two main categories of competitors. There are competitors who use a chemical reaction to capture cancer cells, which is called an antibody-based system. Cell search is one of those. The problem with antibodies is it only works if there's a cell surface marker on the cell, and other than three main cancers, colorectal, prostate cancer, and help me? Breast cancer. They haven't been able to prove that their system works for the other cancers. And secondly, the individual cells in the bloodstream, the metastatic ones, the mesenchymal ones, don't have the cell surface markers. And that's why the antibody systems didn't work in ovarian cancer. They don't work for that. And secondly, there's a membrane-based technology. There's a group of companies who have membrane technology where they set a pore size to capture the cell based on size. That's a really good approach, actually, and it does capture quite a lot of cells. Problem is that the cells get stuck inside the pore. They can't then harvest them out for analysis. So it means that you can't use it for the key application that you want to do.
I'd probably add as well at the moment that there's another area which is called cell-free DNA, and that's slightly more advanced and developed in where it is in getting into the market, and people such as Illumina are using next-generation sequencing on that. But that has its problems, one of which is it only provides an answer based on DNA, whereas if you've got a CTC, you can also do RNA analysis and protein analysis, and the markers that have been developed by Vienna, for example, are all RNA, and that's where we're getting some very good results.
Any other questions? Gentleman here first, please.
Obviously, getting FDA approval would be very important for the success of this company. what is the likelihood, or what would be the biggest reason for the FDA to deny approval?
There's a possibility they would deny approval for the platform that we're going for. So because we're saying you can analyse the cells for any purpose, they might consider that to be too broad. And they have already told us, in fact, that we originally went for a submission of any kind of cell coming from blood, and they've said that's too broad, you need to zero it down on one or more types of cancer, and we've agreed to do that. So there is a possibility they might say the breadth of analytical application is too wide, we're not satisfied with that. We also have a parallel track program, which is, as I mentioned, the ovarian cancer application, which is specifically triaging patients before surgery. So that clinical study is being designed specifically to enable FDA regulatory on a very, very specific application. We'll do something similar in breast cancer and colorectal. Our plan, however, is to get the platform authorization and then build on top of the platform specific authorizations, and that will give us a big step forward. But we've specifically designed the other work in such a way that if the platform thing was some reason we didn't get it, I mean, obviously I'm hopeful we will get it later this year, but if we didn't get it, it doesn't hold back the specific applications.
And I think I'd add as well that the specific applications help get reimbursement in the US.
I think we had one final question at the back.
Sorry, I just wanted to ask, currently with your sample you then discard it. Is there any chance that you could auto-transfuse it and pass it back to the patient?
I didn't hear the question.
Further down the line, yes, you could do that, but
And how quickly can you do that?
well, at the moment, that's not something we're focusing on. I mean, it's in theory you could do it. We don't think that necessarily be the smartest thing, because I think if you look at it, what you want to do is get a drug that's going to target the actual cancer and kill it at source. I mean, just if you keep on taking them out of the body, there's still going to be some residual. The chances of you getting everything out is slim. So,
Well, they're being shed all the time, so you clear them out, and an hour later you've got some more.
yeah.
So it's very, very difficult to strain them out. And I mean, there are ways ideas of doing dialysis, for example, and that's probably what you're talking about. And that could have some medical benefits, but it's got more challenges for getting much more challenges for getting approval than what we're already doing, to be honest. So.
Okay. Gentlemen, I think we're out of time on that one, so thank you very much for Andrew and Ian.